I'm Dr. Professor Mehrotra. I have done my Mbbs from GSPM Medical College, Kanpur, MD in Clinical Microbiology from King George Medical University, Lucknow, and postdoc from PGI, Lucknow. I am teaching as Professor Head in Department of Pathology and Microbiology in various institutes of Medical and Dental Sciences in Lucknow. And I have teaching experience of more than 25 years in medical field. I am also a practicing microbiologist. So today we will start with a chapter known as complement, and at present I am working as faculty in the department of PG department of Clinical Microbiology, TS Mishra Medical College, Lucknow. So today's topic is complement system. As the name indicates, complement हिंदी में कहें तो बढ़ावा देना, बधाई देना, प्रोत्साहित करना, तो वही कॉम्प्लीमेंट का वर्ड यहाँ भी यूज होता है. कॉम्प्लीमेंट यानी कि ये इम्यून सिस्टम में एक ऐसी प्रोटीन्स होती हैं जो एक दूसरे को सहारा देती हैं और सहारा देते देते नींव से पहली मंजिल और दूसरी मंजिल तीसरी मंजिल लेके बड़ी बिल्डिंग बना देती हैं. So complement are the group of plasma proteins, about 30 plasma proteins, which plays very important role in our immune system. So immune system, IMMUNE. So the complement word is derived from an English term and it helps in our innate immunity. Now we will see the history part of it. What happens in 17th century, mice blood was injected in a petri dish in a well, and this mice was resistant to Vibrio cholerae. So its blood was kept in a petri dish and the Vibrio cholerae colonies were grown. And after incubation, it was seen that this blood doesn't allow the growth of the Vibrio cholerae colonies, or the Vibrio cholerae colonies were killed by this blood. Now in the second experiment, the blood is being heated, and when the blood was heated, now it is again put into the petri dish, and the colonies of Vibrio cholerae were grown on the petri dish by inoculation. It was incubated. Next day, it was seen then. This time the colonies of Vibrio cholerae were present. This means the heated blood doesn't give protection to the cholera bacteria. So two types of components must be present in this type of blood, which is found in a guinea pig or a mice which is resistant to cholera bacteria. मतलब कहने का ये है कि गिनी पिग में या माइस में दो तरह के कॉम्प्लीमेंट्स थे. एक तो हीट से डिस्ट्रॉय हो गया, एक हीट से डिस्ट्रॉय नहीं हुआ. जो हीट से डिस्ट्रॉय हो गया है वो उस वर्ड को कॉम्प्लीमेंट कहा गया. किसने उस वर्ड का कॉइन किया? जूल्स बॉर्डेट ने. और जूल्स बॉर्डेट ने कॉम्प्लीमेंट नाम दिया इसको और कॉम्प्लीमेंट कैस्केड की डिस्कवरी की. यानी कॉम्प्लीमेंट कैस्केड वाज डिस्कवर्ड बाय जूल्स बॉर्डेट. लेटर ऑन पॉल अर्लिक said that the heat stable component in the blood was antibody. So जब हीट करते हैं ब्लड को तो जो डिस्ट्रॉय हो जा रहा पार्ट वो कॉम्प्लीमेंट है, जो चीज डिस्ट्रॉय नहीं हो रही है वो एंटीबॉडी है. इसीलिए ये दोनों कंपोनेंट्स हमारे ब्लड में रहते हैं और हमारी इम्यूनिटी को गवर्न करते हैं. सो कॉम्प्लीमेंट कोई नई चीज नहीं है ना. कॉम्प्लीमेंट इज फाउंड इन इनवर्टिब्रेट्स, वेरी लोअर एनिमल्स ऑल्सो लाइक मॉस्किटो, सी अर्चिन्स, starfishes, and they play very vital role in their defense. So it is not a new thing in human beings. It is also found in invertebrates. Now the complements are formed mainly in the liver, or they are also formed in some other minor sites like monocytes, macrophages, cells of the gastrointestinal tract, and cells of the genitourinary tract. Now, properties of complement. Number one, there are 30 group of plasma protein which with the Fc fragment of the antibody. Third, they are destroyed at 56 degrees centigrade when heated. And the major part where they are formed is liver, which is the second largest organ of our body after skin and the largest organ of our body. In complement system, there are three wings which play very important role in complement cascade. One is complement components. The main one are C1 to C9. Second one is properdin system, and third one is regulatory protein. Now, these three things we will tell you in coming slides. So, first of all, the complements are the proteins which are found in our plasma, and they are in inactivated stage. And when any microbe invades our body, they becomes activated. Now, from inactivated stage to becoming activated is a complement cascade. So, how the complement cascade is activated? There are three ways. Number one, antigen-antibody complex when it forms. This is classical complement pathway. So when there is formation of antigen-antibody complex, classical pathway comes into play. Second, when straight away the bacterial endotoxin comes in our body, then alternative pathway starts. And the third one is lectin pathway, where lectin of the host cell combines with the mannose sugar of the microbe to start this lectin pathway. Now, first is the classical pathway. Though it is a classical, but when a microbe comes in our body for the first time, this classical pathway is not initiated. The pathway which is initiated is alternative pathway. This is very important. But when antigen-antibody complex is formed and second time the antigen is exposed, then antigen-antibody complex forms and the classical pathway starts after some time. So initially starts is alternative pathway, and later on the complement cascade which is started is antigen-antibody reaction stimulus leading to classical pathway, and the third one is lectin pathway. So when the complement is activated, two types of proteins are formed, A and B. B is a larger fragment and A is a small fragment. A is also known as anaphylactoxin, which play very important role in anaphylaxis and inflammation, and the B part is used in the complement. Now, in all the three pathways, classical, alternate and lectin pathway, four things occur. First is initiation of the pathway. Second is formation of C3 convertase, which is different in all pathways, lectin, alternate and classical. Third is formation of C5 convertase, and the last is formation of membrane attack complex. This is the final product in all the pathways known as MAC, which creates pores in the bacterial membrane. The pores are of 10 nanometer, which allows the water and other micromolecules to go inside the microbial cell, and the microbial cell burst, leading to lysis of the bacterial cell or microbial cell, and thus we get protected against the infection. So first we will start with classical pathway. Classical pathway, as the term indicates, starts with antigen-antibody complex. So antigen-antibody complex combines with C1q, the fragments of C1R, C1q, C1R and C1S. So this antigen-antibody combines with C1q, C1R, C1S and leads to formation of activated C1 with QRS. Now activated C1QRS breaks down C4 into C4A and C4B. C4A, I already told you, this is anaphylactoxin, goes into the anaphylaxis and inflammation part, and C4B is again act on this C2 complement and breaks down C2 into C2A and C2B in the presence of magnesium ion. Now C2A is an exception here. C2A plays important role in the complement cascade, while other A part goes into anaphylaxis and inflammation. So now the reaction with C2A and C1,4B occurs, leading to the formation of C3 convertase. So what is C3 convertase? C1,4B2A, which converts C3 into C3A and C3B. C3A goes into anaphylaxis again, and C3B binds with activated C1,4B2A to form a new complex known as C5 convertase, and this is C1,4B2A3B. Now this C5 convertase acts on C5 again, and C5 is broken down into C5A, anaphylactic toxin, and C5B. Now this C5B combines with C678, leading to the formation of C5B678, supported by C9, which drills the microbial membrane and forms membrane attack complex. So membrane attack complex is C5B, C6, C7, C8, C9, which are causes of formation of pores in microbes, leading to lysis of cell. Now we come to alternate pathway. Alternate pathway doesn't require C1, 2 and C1. They require 3 complement proteins, factor D, B and properdin. This is initiated directly by the microbe. This is the polysaccharide or lipopolysaccharide layer of the outer membrane of gram-negative bacteria especially, and it is directly started as soon as the LPS is exposed or as soon as the bacteria enters the human body. So factor B, what is factor B? It is acute phase protein of 764 amino acid. It circulates in inactive forms, and these inactive forms are known as zymogens, which is activated in alternate pathway and do form C3 convertase. Factor D cleaves factor B only when active form of C3 and C3 water is present, and properdin is a glycoprotein. So alternative pathway is triggered by lipopolysaccharide of the gram-negative bacteria or LPS or yeast cell wall oblique cobra venom factor. So in alternate pathway, endotoxins which are foreign to the host cell activates factor B, and this factor B is broken down into factor BA and factor BB, which combines with C3B. BA goes in inflammatory process and the factor BB combines with C3B, leading to the formation of activated C3BBB. Now this activated C3BBB has a half-life of 5 minutes, so it is very unstable, and it is stabilized by properdin, and the half-life increases to 30 minutes. Now this combines with C3B, leading to the formation of C5 convertase. So in alternative pathway, the C3 convertase is C3BBB, and which combines with C3B to form C3BBB3B, which is C5 convertase. And again, the C5 convertase breaks down C5 into C5A and C5B. C5B reacts with 678 with the help of C9, forms a MAC and created pores, leading to lysis of the cell. Now comes the lectin pathway. Lectin pathway, here the lectin protein is present in the host cell, and mannose is a sugar which is present in the microbe. So host lectin protein is mannose binding lectin here, and aqueous phase reactant is similar to C1q. So this means that the MBL in lectin pathway is similar to C1q of classical pathway. Another host protein, MBL associated serine protease. This is serine protease of host cell which cleaves peptides into proteins, and this is MASP, M-A-S-P, MASP. So MASP and MBL forms MASP MBL complex and changes C4 into C4A, which goes into an inflammation, and C4B. C4B now binds with C2, or C2 is again changed into C2A and C2B. C2A, I already told you, plays a very important role in this complement cascade. So C2A is used up, and there exists MBL, MASP, which is already there, with C4B here and 2A. This is known as C3 convertase of lectin pathway. and this combines with C3b, leading to MASP C4b2a3b formation, which is C5 convertase. And this C5 convertase again breaks down C5 into C5a and 5b, 6, 7, 8, combining with C8 and later on C9 to form pores in microbial membrane. So water goes in and the cell dies of microorganism. Now, this is very important. Neisseria gonorrhoeae. In Neisseria gonorrhoeae, what happens? In Neisseria gonorrhoeae, what happens? Complement play very important role in prevention against gonorrhea, but in certain resistant strains of Neisseria gonorrhoeae, the Neisseria strain undergoes non-covalent bonding with membrane attacking complex, and the MAC is not allowed to create pores in the membranes of Neisseria gonorrhoeae, leading to formation of resistant strains of Neisseria gonorrhoeae. So this is the first example how Neisseria gonorrhoeae escapes complement and resistant strains are created, leading to diffuse spread of gonorrheal infections to other parts. Similarly, Pseudomonas aeruginosa produces elastase enzyme, and anaphylotoxins like C3A and C5A are inactivated by these elastases, and Pseudomonas get resistant. So our tissues can't destroy Pseudomonas in many conditions. So Pseudomonas aeruginosa becomes a resistant bacteria. There are also some other bacteria which can get resistant in our human tissues, like E. coli and Salmonella. Here what happens? That the side chains prevent insertion of MAC in the bacterial membrane, and so it can't create pores, and resistant strains of E. coli and Salmonella are formed. This was about examples of resistance created against complement cascade. Now we come to gram-positive bacteria. Previously we were discussing all gram-negative bacteria. In gram-positive bacteria, what happens? There is a thick layer of peptidoglycan. This means gram-positive bacteria have a thick cell wall, so thick peptidoglycan layer is there. So this capsule provides a physical barrier between C3b deposited on bacterial membrane and CR1 on phagocytic cells. So this physical barrier doesn't allow MAC to create pores, and leading to the formation of resistant Streptococcus pneumoniae microbe. There are other microbes also which evades complement system by inhibiting the complement cascade, like bacteria, virus, fungi, and protozoan. Example is Vaccinia virus, herpes simplex virus, Epstein-Barr virus, Trypanosoma cruzi, and Candida albicans. Now, this is the nutshell example of all the pathways: classical, lectin, alternate. Classical initiated by antigen antibody complex, lectin MBL. Alternate pathway directly by the pathogen, leading to the formation of C3 convertase, which breaks down C3 into C3A. C3A goes into the inflammatory process, and C3B converts C5 into C5A by the help of C5 convertase, and MAC proteins or MAC complex macromolecules formed, which already we have discussed, leading to pore formation in the microbial membrane. Now, there are many other complements also which play important role in inflammation. For example, opsonization. Opsonization means that when a microbe enters, there are some complements and chemicals which gets coated on the microbial surface, and the microbial surface becomes very tasty. That is why the phagocytic cells moves more fastly towards them and do phagocytosis. So this is known as opsonization, and the fast movement or the movement of the phagocytes towards the target cell is known as chemotaxis. Now there are examples of these segments, like degranulation of mast cells in basophil is done by C3A, C4A and C5A. Degranulation of eosinophil is done by C3A, C5A. Extravasation in chemotaxis is governed by C3A, C5A and C5b67. Aggregation of platelets is done by BB complex. Complement which attracts most of the neutrophils or which is most chemotactic is C5A. Maximum opsonization properties of complement is seen in C3b. So these are very important segments which are very often asked in multiple choice questions. Now we come to the complement fixation test. In this, the reagent antibodies unites with the detecting antigen in the presence of the complement. To simplify it, let's take a serum. Here, this is the serum, and this serum, for example, let's consider this serum has no antibodies against any disease. For example, syphilis. This is a complement fixation test of Wassermann reaction is used for syphilis. So blood is taken, it is heated at 56 degrees centigrade to destroy its own component, and it has no antibodies. So we add a complement which is obtained from the guinea pig, and if there is no antibody, no antigen antibody complex will be formed, and the complement which we have just added remains free. And as soon as we add the hemolytic system known as Mbbs, which is obtained from anti-sheep RBC of rabbit blood, gets hemolyzed. And as soon as the RBC gets hemolyzed, total red color is seen in the test tube, and this means the test is negative. On the contrary, if, for example, the test is positive, antibodies are present in the serum. So patient serum is heated at 56 degrees centigrade for half an hour, and the antibodies are present. We add the antigen. The antigen comes from the cardiolipin antigen from the beef heart. Antigen antibody complex is formed. Then we add guinea pig serum, which is complement, and antigen antibody complex combines with complement, fixes the complement, and then soon when we add the hemolytic system, Mbbs, anti-sheep RBC against rabbit blood, this is not hemolyzed, and a button formation is formed, and this means the test is positive. This is known as complement fixation test, which is expressed in the minimum hemolytic dose. The unit is MHD. So what is MHD? 1 MHD of Mbbs is considered to be higher dilution of inactivated Mbbs, which can lyse 1 unit volume of sheep RBC in presence of complement under 37 degrees centigrade, taking 30 minutes with addition of sodium chloride and magnesium. This is just an academic definition. So complement is very useful in clinical practice. We prescribe a complement test for many patients for the diagnosis of various diseases. For example, the most important disease is systemic lupus erythematosus, very common autoimmune disease, a connective tissue disorder in which the C3 is decreased, C4 is more decreased. MCGN type 1, this is a glomerulonephritis, C3 decreased or normal, C4 more decreased. Similarly, this is a full chart showing in which disease C3 is decreased and C4 is decreased or remains normal. So there are many other conditions where the complement protein plays very important role as a diagnostic criteria. For example, immune complex disorders, SLE, the complement protein which is affected is C124. In susceptibility to encapsulated bacteria, the complement plays important role is C3, factor I. Susceptibility to Neisseria C5 to C9 complement plays very important role. and paroxysmal nocturnal hemoglobinuria. In this, the DAF or decay accelerating factor is involved or get deficient. So in this PNH deficiency in the proteins that regulate complementary activity can cause equally serious disorders. Example is PNH, in which the RBC are lysed and there occurs hemolytic anemia and the patient passes blood in urine in the first urine after the sleep. So that is why it is known as PNH. Now, so in nutshell, we have studied the complement, what is complement, what are the properties of complement. There are three pathways: classical pathway, alternate pathway, and lectin mannose binding pathway. How the complement cascade goes on, which type of deficiencies was found in different sorts of disorders. And before ending, let's have some MCQ questions for the PG entrance. Now, for example, one question: of the following component, which one is the most important option in? The alternatives are C1, C3b, C5a and C5b. So the most important option in is C3b. Second question: of the following complement, which one is the most potent in attracting neutrophils? I have already told you the answer is C5a. Next question: of the following, which one is the important function of MAC? So everybody knows MAC proliferates bacterial cell membrane. Next question: a deficiency of which of the following component predisposes to the bacteremia caused by Neisseria gonorrhoeae? The answer is C5b, C6, C7, C8 and C9. So deficiency of these complement lead to resistant Neisseria gonorrhoeal infection leading to bacteremia. So this is all about the complement. Thank you, students. We will meet in the next lecture very soon. Thanks.